In the previous video, I talked about the special group of genes called the cancer-regulating genes. These are genes that normally help our cells function properly, but can trigger cancer when they are mutated. There are two types of these genes in our cells: cancer-promoting genes and the cancer-suppressing genes. I also mentioned that certain viruses carry cancer-promoting genes as well. Then, how did the scientists discover the existence of such genes? Among the three types of cancer-regulating genes, the viral oncogenes were the first to be discovered before the ones found in our own cells. More than 100 years ago, in 1911, an American scientist named Francis Peyton Rouse published the groundbreaking and the historic set of experimental results. The experiment was performed using a chicken with a type of cancer called sarcoma. Dr. Rouse removed the tumor, ground it up, and filtered it to create the tumor extract. He injected the extract into a healthy chicken, and remarkably, the same type of cancer developed in the recipient. This result was surprising because at the time, no one really understood what caused the cancer. Most people simply thought it was a disease that happened by bad luck. In this context, Dr. Rouser's experiment, which clearly demonstrated cancer could be triggered by something transmissible, fundamentally changed the way people thought about cancer. So, what exactly in the tumor extract caused the cancer? Further investigation revealed that it was a virus present in the extract. Therefore, chickens infected with the virus, rather than the tumor extract itself, also developed sarcoma. This virus was later named Rouse sarcoma virus, RSV, in honor of Dr. Rouse. For his groundbreaking discovery that a virus can cause cancer, Dr. Rouse was awarded the Nobel Prize in Physiology or Medicine in 1966. Then why does RSV cause the cancer? It was later discovered that RSV carries a gene called SARC, and introducing SARC alone without the rest of the virus was enough to cause cancer. Therefore, SARC became the first cancer-promoting gene or oncogene ever discovered. This raised another important question. Does this gene also exist in our own cells? The answer was yes. In the mid-1970s, scientists found that SARC is also present in normal cells. So, to distinguish the viral and the cellular versions of SARC, they were named VSARC and CSARC, where V and C stand for virus and cell, respectively. Now, a natural question was whether CSARC could also cause cancer. However, unlike VSARC, injecting CSARC into cells did not result in cancer. This led scientists to take a closer look at the two versions of SARC, and they discovered that VSARC is actually a mutated form of CSARC, and when they introduced the same mutation into CSARC, it did cause cancer. This made it clear that although CSARC normally helps cells function properly, a mutation in this gene can turn it into a cancer-promoting gene. This was the first strong evidence that cancer can be triggered by mutations in specific genes already present in our own cells. Thus, CSARC became the first cellular oncogene ever identified. This discovery was a major breakthrough in our understanding of cancer, and for their pivotal contributions to this theory, Harold Varmus and Michael Bishop were awarded the Nobel Prize in Physiology or Medicine in 1989. After discovery of CSARC in 1970s, a natural follow-up question emerged. Are there other oncogenes besides CSARC? And if cancer-promoting genes exist, could there also be genes that suppress cancer? Today, we know the answers to those questions, as I briefly mentioned at the beginning of this video. So far, many oncogenes and tumor suppressor genes have been identified. So, from the next video, we will begin exploring some of the most well-known cancer-regulating genes, especially focusing on how they work and how they've changed the way we understand and treat cancer. Thank you for watching.